So, good morning and welcome to the second day of Hydromedit. I am Vasilis Trigonis. I'm an assistant professor at the Department of Oceanography here in Mytilene, and I will be chairing this session alongside Mr. Loukovitis. It is a hybrid session, so I am actually here along with my colleagues in the Department of Geography in Mytilene, and let's start. I'm gonna follow the program. Just to let you know that I cannot see in real time the various participants in this Zoom session, but I have my colleague here that can inform me, if a speaker is out or anything happens like that. So, welcome, good morning, and let's start, please, with Mrs. Nikiforidou, with the oral presentation on Wenthlake relationships of Trachinus draco and Trachinus radiatus in the Aegean Sea. So please, the floor is yours. Thank you.
Thank you very much. Can I share my screen?
Absolutely, yeah.
Okay. Can you see the presentation?
Yes, everything is fine.
Right?
Yes,
nice.
Thank you.
Okay, hello everyone, good morning. First of all, congratulations to all organizers for this event. My name is Vasiliki Nikiforidou. I'm currently working as a research assistant at the Institute of Marine Biological Resources and Inland Waters at the Hellenic Center for Marine Research. Today I will present you our study with the title Weight-Length Relationships of Trachinus draco and Trachinus radiatus in the Aegean Sea. The greater weaver, Trachinus draco, and the stary weaver, Trachinus radiatus, are demersal fish species that belong to the family of Trachinidae. They can be found in the Mediterranean, Black Sea, and the Eastern Atlantic Ocean from a few meters to 150 meters depth. They inhabit sandy and muddy bottoms, and weavers are mainly captured as bycatch species by bottom trawls and gill nets in the Mediterranean Sea. They are regarded as fish with low commercial value, which can explain the lack of deep knowledge of the biology and ecology of these two species. For Trachinus draco, published information includes several studies on the total weight, total length relationship of the species in various areas of the Mediterranean Sea. While for Trachinus radiatus, a few published information is available on total weight, total length relationship, and there is no study based on the eviscerated weight, and there is only one study about the separation of two sexes regarding their total weight, total length relationship. The objective of the present work were to study the weight-length relationship for the total and eviscerated weight of Trachinus and Trachinus radiatus for each sex separately for the first time in the southwestern Aegean Sea. Differences in these two relationships, I mean total weight, total length relationship and eviscerated weight, total length relationship, between the two sexes of each species were also examined. With this study, we aim to provide essential scientific information for the biology, population dynamics, and fisheries management of the two species. Samples of Trachinus draco and Trachinus radiatus were collected from the experimental bottom trawl surveys carried out in the southwestern Aegean Sea, as we can see in this figure, in stations with depths ranging from 69 to 127 meters in May and June of 2015 in the framework of the HCMR project Epilexis. The total length and total weight of the specimen were to the nearest centimeters and grams, respectively. Macroscope examination of gonads was performed to identify the sex of each specimen. After removing the splanchnic organs, eviscerated weight was also recorded to the nearest gram for each individual. In total, 62 females and 27 males of Trachinus draco were examined, the total length of which ranged between 13.7 with 30.9 centimeters and 13.3 with 26 centimeters respectively. For Trachinus radiatum, 35 females and 41 males were examined. Their total length ranged between 21.3 with 43.2 centimeters and 16 with 38.5 centimeters respectively. Weight-length relationship were estimated based on these two exponential equations for the total and the serrated weight respectively, where A is the intercept and B is the slope of the regression. I mean the Greek A. For each species and sex, the isometric growth null hypothesis was examined with using the student t-test. ANCOVA was performed to investigate the between sexes differences in weight-length relationship with significant level A equal to 0.05. For the latter analysis, in order to avoid bias because of differences in the size composition of the two sexes in each species, the same total length range for the two sexes of each species was used. For Trachinus radiatus, it was this range, and for Trachinus draco this, and radiatus this. Kolmogorov-Smirnov test was applied to ensure the consistency between the length frequency of female-male samples of each species, and no differences was identified in both species, as we can see for these two values of p-value. The parameters of weight-length relationships of Trachinus draco for the total and eviscerated weight are presented in this table. In Trachinus draco, according to the value of the student test, weight-length relationship for each sex revealed a positive allometric growth. No statistically significant difference was defined in the weight-length relationship between females and males in both total weight and eviscerated weight analysis. Similarly, the parameters of weight-length relationship of Trachinus draco for the total and eviscerated weight are presented in this table. Weight-length relationship showed isometric growth for males in both analyses for total and eviscerated weight. For females, positive allometric was defined in the case of total weight, while isometric growth was detected for eviscerated weight. No statistically significant difference was defined in the weight-length relationship between females and males in both total weight and eviscerated weight analysis. The results of the present work indicated differences in the growth type of the two congeneric species, with Trachinus draco indicates positive allometry and Trachinus draco reveals isometry. This seems to be related to the different somatic type of the two species, with a small-sized Trachinus draco growing more in weight than in length, while the large-sized Trachinus radiatus growing in weight as in length. Moreover, the difference in the growth type of Trachinus radiatus using total length or eviscerated weight showed that weight-length relationship analysis in this large species is affected by the splenic organ's weight, which should be considered when weight-length relationship is investigated. It should be mentioned here that the sample period when this work was carried out coincided with the reproductive period of both species, something that these are unpublished data. But the fact that may have affected the gonad weight of the large-sized Trachinus radiatus and consequently the total weight of the species. However, this difference was not found in the case of Trachinus draco, which is a small-sized species with smaller gonads than Trachinus radiatus. The slope of weight-length relationship for both species in the present work was within the range of the values published in the literature, except in the case of B for total weight in Trachinus draco. However, the slope B of weight-length relationship for eviscerated weight in this species was included in the range of published B values. Therefore, it can be suggested that the findings of this research are consistent with those of other areas in the Mediterranean water. Our findings showed no differences in weight length relationship between the two sexes for both species. Ak and Zheng 2013 found differences in the slope B of weight length relationship between females and males of Trachinus draco, but they mentioned that the two sexes differed in their size composition. Numerous factors, including genotype, habitat variations, length composition, preservation techniques, health, stomach conditions, diet, sex, maturity stages, sampling season, growth rate, and age, are attributed to variations in the parameters of weight length relationship. Thank you for your attention. And I'm here for any questions.
Okay, thank you, Vasiliki. Thanks for the very concise presentation and for staying well within the 10-minute mark. So thanks very much. Are there any questions from the audience on this work? Okay, I don't think there are. But Vasiliki, please stay with us. We're staying on topic. If it's okay with you, we can proceed with the next work.
Yes, of course.
Again by Nikiforidou, Anastasiopoulou, and Mytilineou. Again for Vasiliki. Oh yeah, for all co-authors. And it's the wavelength relationship of Scomber-Colias and Drachurus in the Ionian Sea. So Vasiliki, the floor is yours. Go ahead, please.
Well, now I will present you our study with the title Weight-Length Relationship of Scomber-Colias and Drachurus in the Ionian Sea. The Atlantic hub mackerel, Scomber-Colias, and the Atlantic horse mackerel, Drachurus, are benthopelagic species that belong to the family of Scombridae and Charanchidae, respectively. Scomber-Colias occurs in the Atlantic Ocean, Mediterranean, and Black Seas at depths up to 300 meters. Drachurus inhabits Western Atlantic and Mediterranean Sea up to 150 meters depth. Both species are primarily captured by pelagic and bottom trawls, as well as pure science and are two resources with medium commercial value in the Greek waters, while research on the biology and ecology of both species in the Ionian Sea is quite limited. A few studies have been carried out on the weight-length relationship of Scomber-Colias in the Aegean and Ionian Seas and the Strait of Sicily, and several works for Drachurus in the Aegean Sea and the Southern Mediterranean. The objective of the present work was to study the weight-length relationship of Scomber-Colias and Drachurus in the Ionian Sea over the last decade, from 2013 to 2022, and investigate potential trends. This scientific information is necessary for the biology and population dynamics of the two species, as well as in fisheries management. Samples of Scomber-Colias and Drachurus were collected in the Ionian Sea in the framework of the Greek Fisheries Data Collection Program from 2013 to 2022. In total, 702 individuals of Scomber-Colias and 1,026 individuals of Drachurus were examined. The total length was recorded to the nearest centimeters and the total weight to the nearest grams of each specimen. The weight-length relation was estimated based on this exponential weight-length relationship equation, where A is the intercept and B is the slope of the regression. For each species, the isometric growth null hypothesis was examined using the student t-test for each studied year. In addition, the trend of the slope B in the weight-length relationship of each species over the studied period was examined by using this linear regression, where Psi is the slope B of weight-length relationship each year, He is the year, A capital is the intercept of this, the lateral regression, and B is the slope of the lateral linear regression too. The significance of these relationships was based on the p-value of these linear regressions. Difference was considered the significant levels A equal to 0.05. In order to avoid the effect of differences in the size composition of each species, each year and to ensure the consistency and representativeness of the examined samples, firstly, the same total length range of each species was used for each year. Secondly, the samples of each year were derived from the same months outside the breeding season of each species in the Ionian Sea. And thirdly, the frequency of all samples were pairwise compared using Kolmogorov-Smirnov test. No significant differences were detected in the length frequency between the samples of all years for each species. The parameters of weight length relationships of Scombrocoleas are presented in this table. Positive allometric growth was detected for most of the studied years, although two cases showed isometric growth for 2018 and 2022. The parameters of weight length relationship of Trachurus are presented per year in this table, respectively. A positive allometric growth was detected for most of the studied years, although two cases showed isometric growth for 2014 and 2020. Regarding the linear regression of the slope B of the weight length relationship over the studied period 2013-2022, although the decline in the B of weight length relationship of Scombrocoleas and an increase in the slope B of weight length relationship of Trachurus was obvious. No statistically significant trend was detected in each species according to their p-values. For Scombrocoleas, this p-value was equal to 0.25. In case of Trachurus was 0.19. The findings from the present study showed positive allometric growth for Scombrocoleas and Trachurus in most of the years of the study period. Furthermore, the lack of statistically significant trend in the slope B of weight length relationship with year for both species may be related to the limited study period, approximately 10 years, of the present work. The majority of the values of the slope B of weight length relationship for Scombrocoleas and all the values B of Trachurus were within the range of the values published in the literature. Variations and differences in weight length relationship parameters are usually attributed to a wide range factors such as this, I said before. And, thank you again for your attention.
Thank you, Vasiliki. I don't know if there are any, I'm repeating again, so that you know I cannot see all the participants in this Zoom session. So I don't know if there is any questions from the audience. Please go ahead. Nothing I can see, Vasiliki. So thank you so much for both.
Thank you too.
For both works, and we continue. We're ahead of schedule with the work. We continue with the work of Danci and Megalofonou regarding the microplastic ingestion by the decapod crustacean Nephrops norvegii in the Aegean Sea. So the floor is yours, please. Thank you.
Good morning.
Good morning.
1 minute. Can you see the presentation?
Yeah, we can see it and we can hear you. Go ahead, please.
Good morning from me. I am Christina Danci, and today I will present my study entitled Microplastic Ingestion by the decapod crustacean Nephrops norvegicus from the Aegean Sea. So, I will begin with some information and characteristics about plastics. Plastic pollution is a significant and pressing issue. Plastics enter in marine environment from anthropogenic activities and tend to be accumulated in marine organisms. There are synthetic polymers in various shapes, colors, and types, and disintegrates due to natural wear, creating smaller particles such as micro and nanoplastics, according to their size. Regarding the species of interest, we studied Nephrops norvegicus, commonly known as Norway lobster or langustina. This benthic decapod crustacean is of high nutritional value and is considered the most important commercial crustacean in Europe. Researchers suggest this species as a good biomarker for pollution and especially plastic pollution due to its low position in the trophic chain, its bottom feeding habit, and its wide geographical and bathymetric distribution. Moreover, Nephrops norvegicus has a gastric mile located inside its stomach, and that is the reason of the formation of the entangled fiber plastic balls in its stomach that stay for longer period also than the other plastic. The aim of the study is to determine the microplastic ingestion in Nephrops norvegicus and to investigate the occurrence, the abundance, and the characteristics of the microplastics that found in the species. We will move now to methodology. Wild population of Nephrops norvegicus were sampled from the North Eboyan Gulf, a semi-enclosed gulf of Greece, Aegean Sea, with high biodiversity and pollution pressures, such as urbanization, industry, agriculture, fishing, tourism, and human activities. A major source of pollution is the mining and metallurgical industry located on the mainland at Larimna Bay, as you can see in the map. The research was conducted in the pointed area here on depth between 100 and 220 meters. The number of the individuals was 203 in total and from all the seasons. After the cuts of the samples, we also recognized the sex and the molting status of the individuals, and we took some morphometric measurements as total encephalothorax length and total weight. We also removed the stomach and we measured the stomach content weight. For the detection of the microplastics in the species, we used the MedSci-Liter protocol according the MedSci-Liter protocol 2019. Accordingly, the stomach content weight transferred in a glass Petri dish, to which we add 20 milliliter of hydrogen peroxide 30% for every gram of sample. After allowing digest for 3 days, the process continued with the filtration of the samples using a Buchner funnel with a glass fiber filter of 47 millimeters in diameter. And also to avoid infections in the process, one blind sample was placed in every set of 10 samples. Additionally, plastic gloves or tools were not used. Laboratory clothing was made of cotton, and all the experiments were conducted in the fume hood. Once the glass fiber filter were dried, I captured the plastics and I record their characteristics such as their type, length, color, and area for the entangled fiber balls using ImagePro Plus software. Statistical analyses were conducted with StatGraphing and SPSS. When it comes to results, microplastics were detected in 161 individuals with at least one plastic particle found inside their stomach content. This accounts for 79.3% of the total sample. Differences found in occurrence between the sexes and the seasons, with a higher incidence observed during the summer season, maybe due to the high tourism, as you can see also in the diagram. Now, the plastic particles that found it was 756 in total, with fibers comprising 90.9%, following by untruncated fiber bulks with 7.1%, and with fragments with only 2%. It's crucial to mention that the percent of the untruncated fiber bulks increased up to 40% in the summer season. Here you can see some images as they were taken from the ImagePro Plus software with the different types of microplastics and with a scale bar. As you can see, some are smaller, some are bigger, the fragments, the fibers, and the untruncated fiber balls, only fixing nets, one color, or multicolors. So the length of the microplastic ranges from 0.09 millimeters to 34.13 millimeters, with a mean length of 3.43 millimeters and a standard deviation of 3.7. Of the plastic detected, 80.2% were microplastics, so up to 5 millimeter, 19.23 were mesoplastics, and only 4 0.57 were macroplastics. Statistical significant differences in the mean microplastic length were found between the sexes and among the seasons. The predominant colors of the plastics were white, black, and transparent, with around 24% each, following by red, blue, and yellow with 17, 3.8, and 3.5% accordingly. Other colors appeared at less than 1%, and the greater lengths were found in the white and red colors. So the abundance of microplastic per individual ranged from 1 to 35, with an average of 3.72 per individual. Statistical significant differences found among the seasons, as you can see, but no between sexes, molting status, or size classes. Also no relationship found between the stomach content weight and the abundance of microplastics. As you can see, also the abundance of microplastics are higher in the summer season. Comparing the abundance of microplastic per individual with other studies from the Mediterranean Sea and the Atlantic Ocean reveals that microplastics abundance is higher inside the Mediterranean Sea, especially in highly touristic zones such as Sardinia or Barcelona bays. In conclusion, the occurrence of microplastics in the individuals was medium compared to other studies. The abundance of fibers were high in the samples, and a significant increase in abundance of the untruncated fibered balls noticed during the summer season, probably due to the increasing plastic pollution of the area. Overall, there is a low exposure to microplastics from consumers, and there is no concern for human health from consuming these species, because the stomach that contains most of the plastics usually discarded and not eaten. Thank you very much for your attention.
Thank you, Christina. Thanks very much. Nice closing slide. And thanks very much for staying within the 10-minute mark. Is there any questions from the audience, either here in the room, Yanis, or from the remote participants? And if not, I have a question, Christina. This is well outside my field of expertise, but I was wondering, you showed close to the end of your presentation a slide for regarding the color of MPs that you found. And I'm just wondering, because I don't know, why is color important? And you you know, you had to show it, and okay, grayscale is more as well as red, and then other colors. So can you please, shed some light on that? Thanks.
Thank you for your question. The color is important to understand the source of the plastic. They have been, there are studies about that. The relationship between the colors and the source of the plastic, because we didn't did until now the FTIR is a method to see the chemical type of the plastic. We are planning to do it. So until now I have only the color, and with the color I try to explain what kind of plastic it is. I believe almost all the plastics were fishing nets. And the color, the red color can be in some studies they suggest that the color is usual connected with what they prefer to eat, the animals. So because nephrops is an animal that eat also nephrops, the same species, maybe the red color can be connected with that. But the white is for sure fishing nets.
Okay, thanks very much for this great answer. I mean, okay, now it makes sense. Thanks very much. Is there any questions? Okay, thank you. And I think we can proceed with the next presentation. I apologize if I mispronounce the surnames. Sorry. Is it Hamdi and Kamanli and Ozak? I'm not sure. Sorry for my Greek-English mix. So we're going to hear about the discovery of Caligus maderensis from the Pompano Trachinotus ovatus in the Mediterranean Sea. So the floor is yours, and please go ahead, share your screen, and thank you very much.
Hello, thank you. Could you see my presentation?
Yeah, but okay, now in presentation mode. Yes, go ahead, please.
Okay, perfect. Thank you for being here. I'm Ilham Hamdi, a doctor from Tunisia. I worked on fish parasites, and today I'm going to present some of my research about the discovery of Caligus madeirensis from the Pompano Trachinotus ovatus in the Mediterranean Sea. So fish parasites are known to cause severe damages to their hosts, as you can see for the group of copepod parasites in the photos. They can attach massively to the teguments and to the gills, causing lesions and hemorrhages, and in extreme cases to the death of the fish. In that case, they can induct economic loss. So the objective of this work is to investigate the copepod parasites of the Pompano fish Trachinotus ovatus in Tunisia for its commercial important value. About 28 fish samples were bought from local fishermen in the Bay of Bizerte, north of Tunisia. During the external examination, the external examination revealed the presence of the copepod parasites from the genus Caligus attached to the tegument and near the pectoral fin, as you can see with the red arrows. The parasitological indices were calculated, and the prevalence was about 14.3%, the mean intensity equal to 1, and the mean abundance was 0.14. Here is the photo of the Caligus species collected under a stereo microscope, but for further identification, we decided to go for the confocal microscopy to see morphological details of the parasites. Here is the maxilla. The first characteristic is the presence of minute spinules at the distal posterior margin. The second characteristics is the maxilliped. The protopodal segment is elongated and unarmed, and the distal segment is about 2 times longer than terminal claw and carrying 2 small setae at base of the claw. The third characteristic is about the exopodal segment of the first leg, which carry 3 plumose setae posteriorly and 4 terminal elements with 3 spines, each carrying an accessory process much longer than the spines, and one inner seta longer than the other spines. The final characteristic was about the exopodal segment of leg 4, which is 2-segmented. The first segment is armed with a long, slender outer distal spine here, and the second segment armed with 3 apical spines increasing in length from outer to inner. So in conclusion, we compared our specimen with 2 Caligus species that were reported from the Pompano fish, Caligus madeirensis from the Northeast Atlantic in Madeira Island, and Caligus mutabilis. Also, we compared our specimen with 12 Caligus species reported from Tunisian marine fishes, and we concluded that the morphological characteristics are conform to Caligus madeirensis. This study also constitutes a new record of Caligus madeirensis in the Mediterranean Sea. Thank you for listening.
Thank you for the work and presentation. Is there any questions? Are there any questions from the audience, please? None that I can see. So thanks very much. Thanks for your work. And I think we can now go to the last work presentation for this session by Pafras Hatziyoannou, Kondylatos, Konidis, Vathidis, Krava, Apostologambru, and Klaudatos regarding the biometric relationships of the rayed pearl oyster Pinctada radiata in the southern Voikos Gulf. So if any of the co-authors are present in this session, please go ahead. Thank you. Okay, I understand my colleague here informs me that Mr. Pafras is going to proceed with the presentation. So go ahead, please. Thank you. Mr. Pafras, can you hear us,
just
please?
a minute. Good morning.
Okay, thank you.
Just a minute, please.
Of course, we're way ahead of schedule, so take your minute and set up your cameras and notes, and whenever you're ready. Thank you. Dimitri, we can see you and your screen. If you can please go into presentation mode and unmute your microphone whenever you're ready. Thanks.
Good morning.
Good morning. Can you switch to presentation mode, please? Thanks.
Yes. I'm ready.
The floor is yours. Go ahead, please.
Biometric relationships of the ray pearl oyster Pictata radiata in the South Evoikos Gulf. Introduction. First of all, the ray pearl oyster Pictata radiata is a marine bivalve viscally attached on soft and hard substrate, many within the sublittoral zone. The species originate from Indo-Pacific Red Sea and the Mediterranean Sea. It was first recorded at Alexandria, Egypt, consisting of a first documented Lesepsianese species in the basin. The Aegean Sea Pictata radiata was first recorded in 1963 from Saronikos Gulf. The aim of present study was to understand pattern of spatial and temporal morphological variability through the assessment of morphometric characteristic from the pearl oyster population of the southern Evoikos Gulf. Material and method. A total of 100 Pictata radiata individual were collected between February 2023 and February 2024 from two sites of southern Gulf of Evoikos, Greece. A range of morphometric parameters was recorded using a dial caliper and image 2 dot blue inside one amaranthos and dot yellow to site 2 γαλάζια νερά. Result comparative box plot of a temporal flash weighting variation of Pictata radiata individual from two sides of Gulf of Evoikos. Volumes are means, red dots, medium horizontal box line, and standard deviation interquate Redditch blocks, and minimal and maximum windscale. General Neal model, result of special temporal effect on flash weighting of the service of Pictata radiata individual from two sides Gulf of Evoikos. Last but not least, the result of present study demonstrates significant spatial and temporal difference regarding the flash weighting of Pictata radiata, where the highest volume were recording during spring and the lowest during autumn. And temporally, the difference is attributed to the productive cycle of the species as analyzed in Musa. Especially the highest difference in flash weighting were exhibited in Site 1, an area with higher organic load to urban descent and associated waste are comparative to Site 2, those more available energy from growth. Similarity is morphometric future exhibit from flash weighting between the population of two investigate sites can be attributed to proximity of these two sites, which less that of nautical mile as seen elsewhere. It is worth mentioning that for spring the means volume of morphometric are higher is our study than those in the later. These differences can be explained by the richer is nutrition coastal water South Evoikos Gulf compared to Maltese water or the fact that EVA costs are more protective by the way dictating action, thus individual can grow larger. The result will contribute to covering part of important gaps in knowledge governing it is description and status invasive species in the Mediterranean Sea and could further contributed its sustainable management. Thank you for attention.
Thank you, Dimitris, for your presentation and for the 5-minute mark on the duration. Thanks very much. Is there any question from the audience, either in the room here or from the remote participants through Zoom? I don't see any, so thank you very much, Dimitris. And this concludes this morning session on the fisheries field and from Mytilene, and also my colleagues here that helped me with the technical aspects, Victoria and Ioannis. Have a great day, and thank you very much. Thank you, Victoria. Thank you, Ioannis. See you. Thank you very much.
Ένα, δύο, ένα, δύο. Ένα, ένα.
Σε ακούμε, Δημήτρη.
Τέλεια, μια χαρά.
Good afternoon from me, and welcome to the second field of fisheries, to the presentations between 12 and 13 o'clock. We will start with Mr. Pafras and the presentation called Socio-Economic Analysis of the South Evoikos Gulf Small-Scale Fishing Industry using Exploratory Data Analysis and Supervised Machine Learning Algorithms. Mr. Pafras, the screen is yours.
Good morning.
Good morning to you too. Can you share your screen, please, with the presentation?
Yes. Just a minute, please. It's okay.
We can see nothing. Why? You think? Just a minute, please. Okay, we can see you now. We can see the presentation. You can start. You have approximately 10 minutes. Okay?
Yes.
We cannot hear anything. Are you speaking? Mr. Pafras, can you turn on the microphone? We cannot hear anything.
Yes.
Okay.
Are you ready?
We are. Are you?
Yes.
Okay, you can start.
A socioeconomic analysis of the Southern Evoikos Gulf small-scale fishing industry using explorative data analysis and supervised machine learning algorithms. First of all, Greece's small-scale fisheries are vibrating part of coastal life with numerous fisheries operating a wide range of boating scattered across the long coastal line. The whiz diver fishing great and danger variety of species, making the fisheries remarkable variety. These small-scale operations are not only crucial source of fresh, high-quality seafood, but also time novel wildlife from many islands and coastal community with deep sea to the sea. The South Evoikos Gulf sweet eating within the area predictive industry as area healthily relating of fisheries. A complete field of 1,950 boats operate out of the South Evoikos Gulf with artisanal vessel making up 19% of this total. The aim of the present study was to assess the socioeconomic characteristic of the small-scale fishing field operating in the South Evoi Coast using exploratory data analysis and supervised machine learning algorithms. Material and method. Data was collected during winter spring of 2023 through personal interviews with fishermen operating in the study area using distractor questionnaires. The questions were close-ended, classified into 3 sections: name fishermen, demographic characteristic, and vessel equipment, and fisher data. Total 15 questionnaires were collected, and data were coded in Microsoft Excel and analyzed using a statistical program Jamovi, and predictive analysis using unsupervised machine learning algorithms was further employed to identify the principal contributing conversing on a choice of fisher groups from fishermen used to orange data mining software. Result figure 1 that gillness are used between spring and autumn with more intensive use the summer month. So the trammer net are used almost all years route, and so that the long line is used all year route with highest frequency for use the summer month. Next figure show that most responsive up to 15% fish near agricultural farms. Prevent species caught in the South Evoi Coast Gulf. The most show Octopus vulgarus, Mudzus cephalus, Pangrus, and Boops. The reason from the preference from the fishing field. According to the best performance model, gradient boosting, the main reason that influencing the small-scale fishing section in the South Evoi Coast region in the show fishing group, the vessel total length, followed by the vessel experience and porto embarkation. Last but not least, the provision of information by commercial fishermen is one the most direct approach to enable the link between fisheries and scientific. The result explained an engine workforce, 16 years old mean age with low education, a similarly Aegean field, and fishing activity years route. The present study indicate that the main reason from the choice of fishing groups primary vessel total length of the followed by the fisherman experience and porto of embarkation. An interesting facing you the admission from the friend of the devoting fisher that regularly fishermen that regularly fish in the proximity of floating cage aquaculture farm within 13 meters. Thanks you for attention.
Thank you very much for the interesting presentation. Are there any questions from the audience?
So, good morning. I don't know. Can you hear? So it's mainly not a question, it's as a remark. As a member of the team, I want to thank Mr. Pafras for his presentation and to say that the socioeconomic data in Greece are very few. We have a lack on socioeconomic. It's an every effort that we make to increase the knowledge on socioeconomic data, with especially with the use of new methods like machine learning. It will be very helpful on our effort to improve the management of fisheries and the better and the improvement of the living conditions of the fishermen and on coastal livelihoods. So this work is on the path of this effort. Thank you.
Thank you very much for the clarification. But I can see from the discussion that the ages of the fishermen is above 60 years old, and there are not any younger ones like to be recruited in these professions.
Yes. This is something that the work and the analysis of Mr. Pafras show clearly. We know from the data that we have collected through the national program of national data collection framework, but from all statistics that all the people that are working in agriculture in general are more aged than the general working force. If we go inside the agricultural personnel, we can see that the fishers have the biggest age average than all the other professions on agricultural sector. So it is a significant problem for fisheries because we do not have new fishers joining the profession. So, and there are a lot of effort to improve their working conditions, because if the working conditions do not improve, you will not have any new fishers. And this is something that cannot be done only by their families. You need a lot of work from the state, but also a lot of work from our community scientists to find tools to improve their daily life, not only their income.
Thank you very much. Are there any questions? So we can move to the next presentation by Mr. Latos, and the presentation called Histopathological Alteration in Callinectes apidus due to Hematodinium parese infections in the North Aegean Sea. So, Mr. Latos, are you online? You have approximately 10 minutes for your presentation. And the screen is yours.
Hello, everyone. Hello, Konstantinos. Can you hear me?
Hello to you too. So,
Konstantinos, can you hear me?
Yes, I can.
Okay.
Can you
Can
share
I start?
Can you do it full screen, please?
Of course. Okay. So, I'm Latos Athanasios, PhD student from Aristotle University of Thessaloniki, and as Konstantinos said, I'm going to represent to you histopathology of the Hematodinium parese infecting Callinectes apidus in North Aegean Sea. Globalization, especially trade and evasions through ballast waters from ships, impacts of climate change, and especially rise of average temperature through years in marine ecosystems, socioeconomical changes through escapes from new culture and ornamental species are key drivers for biological invasions, Critical invasion examples are the crustaceans, which they consist the most successful taxonomic group of aquatic alien invaders. Bioinvasions can interact with the native biodiversity by modifying habitats, changing the community and food web structures. Typical examples are Passifastacus leniusculus from aquaculture, as production from aquaculture, and Calinectes sapidus from ballast waters, which they consist successful invasions. One of the most successful invasions in marine European ecosystems, as I said before, is the invasion of blue crab. The first record of the American blue crab, Calinectes sapidus, from Europe was collected in almost in the 19th century on the Atlantic coast of France. Subsequently, specimens were detected in North Sea, Mediterranean Sea, and Baltic Sea and Black Sea, and possible Sea of Azov later. Ecological consequences include predation for native species, competition for resources, alteration of habitat, and spread of diseases. Economical consequences, such as impact in fisheries due to predation of other species, alongside with damage on fishery nets and bivalve culture threats, can result increase in management cost. Social consequences comprise loss of cultural heritage due to highly predation of the species to the endemic species and change in small-scale fisheries. While invasions can apply pressure to local ecosystems, sustainable utilization of blue crabs as a resource can create opportunities with great economical importance, such as small-scale enterprises with exporting character, or even enterprises utilizing this invading product. Typical examples of utilization of blue crab is the USA. In these charts, we can see the race in the landings alongside with the price of the species. To our knowledge, data on the economics of blue crabs in Greece is scarce, with existing information primarily focused on biodiversity and fisheries. The utilization of blue crabs in Greece began in recent decades, primarily for exporting among the public. Blue crabs are mainly known for the invasive nature and negative impacts in ecosystems. However, their performance was average due to alteration in the population density in its habitats. Mitigation of the population density has primarily been attributed to overfishing, urban pollution, and more generally, the impacts of climate change. However, the species causes of this alteration have not fully investigated over the years. Main objective of this research is to investigate the health status of blue crabs originated from areas experienced mortality events. Moribund and healthy individuals were collected from the lagoon of Porto Lagos in November 2020, originated from mortality incidents obtained from local fishermen. The second and third samplings were performed almost at the same time in November to investigate any potential threat in the local blue crab fisheries in Thermaikos Gulf. We kept them as control. Crabs transported to the laboratory and anesthetized by placing them on the ice for 4 minutes. Oops, I'm sorry. Haired gills and hepatopancreas were selected for the histopathological examination. Half of each tissue placed in the fixative agent, and the second half kept in -20 for molecular analysis. No microscopic signs detected, apart from the lethargic behavior of several individuals. Tissues intended for histopathology stained with hematoxylin and eteogen staining and observed in microscope. The second half of its tissue, intended for molecular analysis, was stored in -20 until DNA extraction. Primers intended for molecular part were selected according to the histopathological results. Histopathological sections of healthy gills of Calinectes sapidus can be seen in the first picture, when you can see the non-infected ones. And in the second column, you can see the infected tissues of crabs with uninucleate and multinucleate stages of the species, of the pathogen species, depicted in gills, pancreas, and heart. In the second picture, you can see the sections of heart in the of blue crab, and especially myocardium and pericardium. You can see, and you can see with arrow and arrowheads the trophons, the filament trophons, and the amoeboid trophons in the heart. Histopathological displays of hepatopancreas can be seen in both A and B pictures. No Hematodinium perezi cells in the first one, in the second one, histopathological changes can be seen in the hemal space of the hepatopancreas, and trophons in multiple forms. Concerning the presence of the parasite in the tissues and in the histopathology in the species, abnormalities were mostly detected in the digestive gland. Specifically, as you can see in the second picture, in the picture B, the concentration of the hemocytes were higher in infected specimens, which is related to the immune responses of the host to this invasion. Additionally, the abnormal development of the epithelial cell walls was detected in infected specimens. You can see the thickness in the walls of the digestive glands. And especially specimens infected with Hematodinium perezi demonstrated, as I said before, thicker cell walls in the tubules, in the digestive tubules also. In the third picture, you can see the histopathological display in gills infected with ciliates as a co-infection with Hematodinium perezi. Confirmation of the species has taken place with the use of primer pairs, and you can see it down on the screen. This research presented a new agent, as we discussed, implicated with the declines of the species. Hematodinium species have a worldwide distribution and long history infected crab species, such as USA, China, Australia. This pathogen is very important and infects various of crustaceans, often leading to substantial mortalities, as in Chesapeake Bay in the USA, mostly in marine crabs, with mortalities of the most important reaching up to 86% of local population. Immediate action is needed to monitor ecosystem and establish new regulation for managing invasive species. Is mainly needed in order to mitigate the impacts of this behavior. Special thanks to all the team that committed in this project.
Thank you very much, Mr. Latos, for the interesting presentation. So, are there any questions from the audience? No? So I have one question. Do we need to provide this strategic management for the invasive species? Because from what I can understand from the presentation, there is a pathogen that keeps the crab species into a plateau. So do we need to recruit these management plans, or let the nature do what it does best?
Well, it depends on what strategy we need to follow on the species. I believe that we can utilize these species in order to obtain and make and product a lot of a lot of works, especially in fishermen. It's an important income for them, but we need also to protect the other resources implicated with these invasions, like mussels. I think Spain has a unique model in order to prevent the increasement in the race of the production of the population of this species by catching them and marking them and catching, I think, only the males and release also in the environment the females. And they have very how can I say it? They have great results in the management of the species with by selecting only the males.
Okay, thank you very much for this clarification. Thank you for being here, Mr. Latos. And we will move on to the next presentation, population structure of the devil firefish, Pterois miles in Rhodes, Greece, in Rhodes Island, by who is presenting? Is it?
Hello.
Okay, I cannot spell the name. Can you do it, please? Okay, okay. So the screen is yours. You have 10 minutes for the presentation. Okay. And you can start.
Okay. You have to make me co-host. I can present. I can share my screen.
You can do it now.
Okay,
yes.
Can you see my presentation?
Yeah, can you do it on full screen, please?
Of course, yes.
Okay, thank you.
Thank you. Hello, everyone. My name is Theodora Karakiaourova, and I would like to present to you part of my undergraduate thesis study that it is about the population structure of the devil firefish, Pterois miles, in Rhodes Island. Let's start with a few words about the devil firefish. So, Pterois miles, native to the Indian Ocean and the Western Pacific Ocean, is an invasive fish species of particular concern, which has established large populations in the indented areas of the Western Atlantic, including the Gulf of Mexico and the Caribbean Sea. In the Mediterranean Sea, the species was first recorded in 1991 off the coast of Israel. In the coastal waters of Rhodes, the devil firefish was first recorded in 2015. Because of its adaptability, reproduction characteristics, and generalist predation, the devil firefish has been rendered the fastest invader of the coastal waters of the Mediterranean basin. As the devastation caused to the ecosystem by its spread in the Atlantic Ocean is well known, its appearance in the Mediterranean is causing concern and management measures must be taken. So the objective of our study was to assess the population characteristics of Pterois mice, namely sexual, spatial, and temporal variations of its biometric characteristics, age composition, and growth in Rhodes Island in order to contribute to a growing literature that will facilitate the sustainable management of the species in the Eastern Mediterranean Sea. Having discussed the background and the objectives of our study, let's now move on to the materials and methods that we employed. Experimental trials were performed monthly from April 2021 to March 2022 in depths between 8 to 35 meters with gill nets and trammel nets, employing 2 small-scale commercial fishing vessels within 3 areas of the coastal waters of Eastern Rhodes, as you can see on the map. In total, 36 holes for each fishing pier were performed and 363 individuals of Pterois miles were collected, measured for total and standard lengths, body depth in centimeters, and total weight in grams. Then sex, spatial, and temporal variations were assessed for all the morphometric characteristics. Length frequency distributions were converted into age classes with Bhattacharya's method. Growth parameters were calculated using the von der Laan growth equation, and maximum age was calculated according to the equation from frozen Bioland 2000 that you can see on the blue box. Okay. First, as for the sex, spatial, and temporal variation, you can see at the tables that there were significant differences for all the morphometric characteristics, except for the body depth between females and males. The results shown that the biggest and heaviest fish for males were found on site 3 and were caught during summer. To study the effect that each factor had on the total length and total weight of the fish, a least squares regression model was employed that showed a significant interaction effect on total length and total weight, first from the season, followed by the fishing areas, and lastly from the sex. Moving on to the Bhattacharya's method, 5 age groups were identified with the third year class exhibiting the dominant cohort, with 41.5% of the total population, where the mean total length was 25 centimeters. According to von Bertalanffy growth equation for the total population, the asymptotic length was estimated at 45.35 centimeters, the K value at 0.18, and the T0 at minus 1.55 years. In the green box, you can see the growth equation for males and females respectively. A higher asymptotic length was estimated for the males. As you can see on the frequency histogram, there were not found females older than 6 years. Maximum age for the total population was estimated at 17.5 years and a much bigger 18.5 for the males. Furthermore, the growth performance index was very similar for both sexes. So, according to our results, it is obvious that seasonality exerts the highest effect on total length and total weight of pteroid mice. The morphometric differences among sexes in our results correspond to males being larger in total length and heavier than females. Individuals collecting during summer were larger and heavier compared to winter, which was also pointed out for Cyprus area. This can be attributed to various reasons, such as fish year selectivity and an androgenic shift of larger individuals to deeper waters out of our depth range during the winter. Spatially, individuals collecting from fishing area 3 exhibited significantly larger total length and total weight compared to those from the other 2 areas, which is possibly attributed to the lowest fishing pressure in that specific area. Our result of the 3rd year's old class was the predominant, contrast the results from a study in Cyprus who identified 4 cohorts with the dominant present in the 2nd year old class. On the other hand, in Turkey, 7 age classes were estimated. In long-established populations in the Atlantic Ocean, the rose mice has exhibited up to 9-year classes. The maximum lifespan of the species is known to be more than 30 years old. However, this value corresponds to an individual kept in an aquarium and most probably does not reflect the longevity in the wild. In our study, the estimated maximum age is 17.5 years for both sexes, which is still a rather long period. Furthermore, the von Bertalanffy growth equation produced an asymptotic length closer to that from Turkey and distant from that from Cyprus. The same trend applies for the K value. Our results indicated that in a short period of time after the first documentation of pteroid mice in Rhodes Island, the species has developed a well-established and well-structured population. The provided information can facilitate modeling population growth in the prediction of rates of expansion of the species in the basin and contributes towards sustainable targeting fishing practices. Additional research that we did on the mortality and reproduction of pteroid mice showed that the population in Rhodes Island is underexploited. This could mean that the devil firefish can be a potential new fishing resource in the area and also all the Mediterranean Sea. After all, if you remove the venomous spines, you can cook it any way you like and totally safe with it. Here are some of our references. Thank you very much for your attention.
Thank you very much. This was a very beautiful and interesting presentation. I don't have any questions for you. I don't know if the audience, someone from the audience does. Okay, so we can move on to the next one. Thank you very much for being with us. So, Mr. Karagiannis, small-scale fishing, what is it, métiers? Καλά το λέω. Ναι, métier, in the inner Ionian archipelago. Karagiannis et al. Mr. Karagiannis is presenting live.
Hello, hello. Is everything okay? Okay. Hello, everybody. My name is Asimakis Nestoras Karagiannis. I'm an undergraduate student in the University of Aegean in the Department of Oceanography and Marine Biosciences. And today I'm going to present to you the work that I did during my internship at IC Environmental Organization, in which we identified small-scale fishing métiers in the inner Ionian archipelago. Κάνω λίγο τα controls. Okay. So, small-scale fisheries, the Mediterranean relies significantly on them. They are a crucial economic driver, and especially in Greece, in which they account for over 97% of all fishing fleet. Even though there is an absence of reliable fisheries data in data-poor fishery areas. Of course, this hampers the evaluation of the state marine resources. The fishing industry in the Greek Ionian Sea, in which our study area today is a part of, is distinguished by a diverse range of species, and the fishers there use numerous fishing gears. That's how the usage of the different fishing gears to target different target species, that's how small-scale fisheries can reflect the complexity of the ecosystem of the fishing, with the difference of the fishing effort in different seasons and in different spatial extents. The Inner Ionian Archipelago, this is our study area for this research. It's a Natura 2000 site. It has been designated as a site of interest in 2006, as a site of community interest in 2006 and 2011, and it was also designated as a special area of conservation. Even though it has been designated as a special area for many reasons, it's also an important marine mammal area, an important bird area, a key biodiversity area, and more recently, it was also designated as an area of interest for sharks and rays. There is currently no management plan regarding fishing activities, and the prohibitions are only limited to fishing with dynamic gears. To gather information about the fishing strategies, a questionnaire was developed, which then IC personnel went to 9 ports in the study region, and it approached the fishermen in a relaxed manner, and they had structured interviews with them to gather information about their fishing strategy. Then the fishers were asked to mark down on a map their fishing spatial extent. The data was then taken back to the office. It was digitized and organized in a database. And through analysis of this data, we were able to identify the métiers, which all this information was then integrated in GIS for further analysis and visualization. That's how we were able to make maps of the spatial and for some of the métiers temporal extent. So 33 interviews were conducted in 2023 in 9 surrounding ports of the area. These 33 interviews covered 10% of the total fishing fleet in the area, according to the EU registry of 2023. And 50% of the responders, they derived their livelihood exclusively from fishing. The data that was collected, it regarded gear specifications, things like mess size, hook size, distance from shore. It got information on the target species, the depth, the habitat, and of course the temporal and spatial extent. This is the table with the métiers that they were identified. For the gears, we use the FAO code. We recorded 7 different fishing gears and 27 target species. This made of 39 combinations, which we then grouped into 12 final métiers. The group happened by taking account the gear specification, the seasonality, the depth range, and the target species. This is the first métier. It's gillnets that were used to target Palinurus elephas, and Pagrus pagrus. They were reported throughout the whole year in depths ranging from 70 to 100 meters with a mesh size of 26 to 30 millimeters. It was recorded in about 10% of the questionnaires. We can see here that there is a spatiotemporal change during the summer and autumn compared to winter and spring, which seems pretty stable around the area of Kalamos, Atokos, and Arkoudi. This is the material regarding gillnets targeting Mulus barbatus, Picaras maris. They were reported throughout the whole year again, using with depths ranging from 15 to 150 meters, with a mesh size of 25 millimeters. They were reported in about 10% of the total questionnaires. We can see that there is some spatiotemporal change here. Of course, it's less prevalent than the previous. These next 3 métiers, no spatiotemporal change, it was recorded for them. The GNS3, which it's a gillnets regarding Merluccius, it occurred about 35 of the total questionnaires with a mesh size at 36 millimeters. Of course, most of the fishers, they recorded that they fish in the same areas throughout the whole year for this specific métier. The GTN1, GTN2, these are the gears which is gillnets and tramelets combined. They are used to target Mulus rumulets and Sarda sarda with a mesh size of 36. This is a GTN1. And GTN2, it's again gillnets and tramelets to target Dentex, Diplodocus arcatus, Poteos, Macantharus with a mesh size of 40 millimeters. These 2 métiers, they were recorded only like 1 or 2 times. So no spatiotemporal change was recorded for them. This is now we're going to the tramelnets. This one, the GTR1, it was the most recorded métier. It was recorded for more than half of the total interviews. It is used for Mulus rumulatus, Picaras maris, Boops, Scorpina scrofa, Oblada meranura, and Serranus cripa, with a mesh size ranging from 24 to 70 millimeters and a depth range from 4 to 70 meters. We can see here that during the first half of the year, winter and spring, the effort is seen to happening in more open waters than in the summer and autumn, where the effort is more limited to coastal areas. These next 3 métiers, they also don't have any spatiotemporal variability. The GTR2, it was the next, it was recorded in a quarter of the total interviews. It was used to target Palinurus elephas, Pagellus erythnus, Pontiliosum macantharus, Dentex, and Pagrus pagrus. Again, most of the fishermen recorded that they fish in the same areas throughout the whole year. The GTR3, which was used to target Cephalus and Mugilidae, was recorded only 1 time, and GTR4, which was again tramelnets for the European hake, was recorded again to very few interviews. This is the métier for the long lines. The long lines, they were used throughout the whole year. We can see the spatial extent is throughout almost the whole region. The hook size that they were used was 9 to 15, and the depths were ranging from 20 to 125 meters. And we also had 1 recording of a fisher using drifting long lines to dry Xiphias gladius at a depth around 150 meters. Trolling beach scenes are not allowed in Greece. That was not always the case. They were allowed until 2022. There are currently less than 100 active licenses. But according to the Fisher Association of Meganisi, there are more than 15 beach scenes in the area. We were able to record 3 fishers in our interviews that they used a specific gear, mainly to target sardines, anchovies, Picaras maris, and boobs. So there is prohibition. Illegal fishing is still a thing in Formicula in Ireland, only a few meters away from caves with seals on them, on top of Posidonia meadows. This is the annual fishing activity. We can see that the southern part of the area seems to be pretty active throughout the whole year. It's a yearly fishing effort in days. So each spatial unit is how many days this area was fished. The southern eastern part seems to be the most active area, as we will see next to throughout the whole seasons, and also the area next to the port of Mytikas. This is the seasonal fishing variability. We can see again here that in the winter, spring and summer, the northern part seems to be more active, while the southern part seems to be more active, while the northern part of Lefkada seems to be the less active fishing area in the region. It was not even recorded during the summer season. So netters exhibited a larger selection than longliners by targeting a greater number of species. Gillnets and trammelnets are used in different ways, with the latter being less selective. Of course, this can explain the diversity in the number of species caught among gillnets and trammelnets. Fishers use trammelnets with very mesh sizes down to 70 millimeters, focusing down to 70 meters, focusing on the moderate-priced Mullus ormuletus, Picaras maris, boobs, and Oblata melanura. Regarding the longliners, they mainly used medium-sized hooked and targeted a variety of Sparidae, Pinnephilidae to depths up to 125 meters. So, similarly to other Mediterranean studies, we were able to identify a lot of different métiers, which we were able then to diversify by the usage of the different target species and the different gears that are used in the area. This is a preliminary work that showcases the potential to increase knowledge with the use of structured interviews in data-poor regions regarding information about small-scale fisheries, and these researches can be used as a baseline for future research efforts and management. I would like to thank all the professional fishermen that participated in the study. I would also like to thank IC for the internship and for giving me a scholarship for this conference. Professor Moutopoulos for assisting in this research, and Dimitra Kiscu for supervising my internship. And of course, this project was conducted in the context of protecting Inner Ionian Archipelago and Formicule Island, which is implemented by IC and funded by the Blue Marine Foundation and Ion Environmental Foundation. Thanks a lot for your attention.
Thank you very much, Mr. Karagiannis. So, are there any questions from the audience? Kyria?
In beach seines or shore seines. As I saw, that kind of fishery is prohibited from 2020. But if I saw well, you said that there are more than 100 active licenses. So how the managers or fisheries department deal with that? Is those fishers working on seasonal basis with other gears, or were just solely involved in shore beach seine? So if they are prohibited to work, how they survive? Do they, switch on some other fishing gears or what? Or maybe this is the question for the professor.
Okay. So if I understand correctly, your question, you're asking me how are less than 100 active licenses while it's prohibited, right? I'm sorry.
I know why it's prohibited, because the same situation we have in Croatia, because we don't have any more derogation, the same as you, because we cannot comply to everything that the EU needs for the working of those fishing gears. But I'm interested just in social economic part of the story, how fishers that previously worked with that fishing gear, what they do now. Is they still in fishing? Because you said that they have active licenses, but what means you have active licenses if you cannot work with the fishing gear? Do they switch on some other fishing gears or what?
So I guess, of course, the ones that they don't resort to illegal fishing with the tool, they change the gear that they use to, of course, adapt to the laws. The ones that, of course, conform with the law and they don't resort to using illegal trolling, because as we saw here, we were able to record 3 fishermen that still use beach scenes, and we were informed by the Fisher Association of the island of Meganis, sorry, that there are more than 15 or around 15. So I guess most of them change the way that they use, but some of them still continue.
Πω, τι κάνουνε, τεχνολογία. Yes, there's a banning of beach chains, okay, from 2002, right? 2002, it was the general Mediterranean regulation for banning beach chains for all Mediterranean. In Greece, there are some presidential decrees that allows some beach chains to fishing in Greek areas, but in 2020, they prohibited all beach chains in Greece. So these vessels are fish with different small-scale gears, trammel nets, mostly, and gill nets. This beach chain fishing that Asimaki shows in the presentation are illegal fishery. Of course, it's not a legal one; it's illegal. And these vessels are not okay. If we want to be right in our words, is not beach chains, but it's small-scale vessels. It used to be beach chains, but of course they don't use this bag, troll type bag. It's small-scale vessels with trammels and long lines. Okay, but the fishing areas that Asimaki shows are some fishing spots with illegal fishery of beach chains. There is a huge problem in this area of illegal fishery because it's Natura area. Some parts are Natura area, and it's illegal to fish by trolling gears. Okay, thank you very much.
Thank you very much. Are there any other questions for Mr. Karagiannis or Professor Moutopoulos? No. So thank you very much for being here in this session of Field and Fisheries.
Hello, good afternoon. We are moving to the next section. We have a small delay. My apologies for that. And the next presentation is the title is Satellite Data as Potential Predictor Variables of Dissolved Oxygen in Marine Aquaculture. The presenter is if it's ready, he can
Good
start.
evening. Can you hear me?
Yes, yes, yes.
Okay, my name is Androniki Dimoudi.
Okay,
Can I
Kira
share my
Dimoudi.
video? Can you see my presentation now?
Yes, if you want, you can switch to. Okay, yes, perfect. Thank
Okay.
you.
Can I start?
Yes, yes, go on.
Okay. Good evening to everyone. My name is Androniki Dimoudi. I am a PhD student in Department of Ichthyology and Aquatic Environment at University of Thessaly in Greece. And today I will present you our research entitled Satellite Data as Potential Predictor Variables of Dissolved Oxygen in Marine Aquaculture. Water quality indicators include physical, chemical, and biological characteristics, which are traditionally determined by field samplings, in situ measurements, and laboratory analysis. Although these methods provide high accuracy, are costly and time-consuming processes, which are only able to provide point estimations of the water quality parameters. The real-time spatiotemporal distribution of the water quality characteristics is almost impossible. On the other hand, for several decades, remote sensing techniques have been widely used for the retrieval of a variety of water quality parameters and can offer data with low cost or freely available data with almost continuous spatial coverage. The sensors used in remote sensing are grouped in spaceborne and airborne sensors based on the platforms in which they are installed, outside or within the Earth's atmosphere, respectively. These sensors can measure the amount of radiation reflected from the water surface and at various wavelengths, which called spectral bands. Some of the water quality parameters, which called optically active, have a clear influence on the water color, while others not have. So their estimation using remote sensing techniques is a difficult task. These parameters include, among others, the dissolved oxygen. The dissolved oxygen levels in marine aquaculture could be affected by the high fish load, the biofouling observed in the nets of the fish cages, the uneaten food, the feces, and the excretions of the cultivated organisms. Although the major part of the uneaten food is consumed by the wild fish, the remaining food combined with feces and excretions enrich the water column and the seabed with organic matter and nutrients. The decomposition of organic matter is done by microorganisms which consume great amounts of dissolved oxygen. On the other hand, the presence of nutrients in aquatic environment could lead to the growth of aquatic vegetation and plankton. These organisms consume high levels of dissolved oxygen during night, and when they die, they enrich both water column and seabed with organic matter, the decomposition of which, as I said previously, requires high levels of dissolved oxygen. Up till now, a common monitoring approach for the estimation of dissolved oxygen in marine aquaculture includes particularly field measurements. So, This study aims to determine the spectral bands of the satellite Sentinel-2 multispectral instrument that could be used as potential predictor variables of dissolved oxygen in marine aquaculture, conducting a regression analysis. For this purpose, the study's framework includes in situ measurements, data from satellite images, and statistical analysis to assess the collinearity issue between the spectral bands of Sentinel-2. In total, 5 criteria were used to detect collinearity, which include pairwise correlation, variance inflation factor, eigenvalue, condition index, and variance proportion. Now, the study area is located at the southeast part of Pagasidikos Gulf in a cove where floating cage farming is operating. There were carried out 2 seasonal samplings, and surface dissolved oxygen measurements were taken from a grid of 24 sampling stations, as you can see on the map, using a CTD. This grid was developed during a previous study in order to cover the whole cove and not only the points where the fish cages are located. After the field measurements, 2 satellite images from Sentinel-2 were used to be as close as possible to the sampling dates and were downloaded through the Copernicus program. At the upper right side of this slide, you can see the 13 spectral bands of Sentinel-2, which cover the visible, near-infrared, and shortwave of the electromagnetic spectrum. The spatial resolution is dependent on particular spectral band and ranges from 10 meters for bands 2, 3, 4, and 8, from 20 meters for bands 5, 6, 7, 8, 12, and 11, and from 60 meters for bands 1, 9, and 10. So, using the GPS measurements received during the in situ measurements, for each one of the satellites from each one of the satellite images that were used, the pixel values coincided with the stations where the dissolved oxygen measurements were taken, and the pixel value for each spectral band was extracted. So, using the data from the in situ measurements and the satellite images, we want to conduct a regression analysis to create a linear model from which we can predict the dissolved oxygen levels using as predictor variables the spectral bands of Sentinel-2. In regression analysis, collinearity or multicollinearity is a data issue that arises if 2 or more predictor variables are highly correlated. Perfect collinearity occurs when 2 or more predictor variables containing exactly the same information, while high collinearity occurs when these predictor variables containing the major part of the same information. Generally, multicollinearity is a phenomenon that observed between the spectral bands of multispectral images in remote sensing. The problems now that can occur when we use collinear data in regression analysis is the estimation of unreliable regression coefficients and thus the creation of unreliable predictive models. Moreover, it's difficult for us to assess the significance of each individual predictor variable on the dependent variable. So, the first criterion used to detect the collinearity problems among the spectral bands was Spearman correlation coefficient. This coefficient ranges from -1 to +1, and the higher the absolute value, the stronger the association between the 2 variables. This coefficient could be used as a first indication of collinearity between the data in regression analysis. In this table, you can see the pairwise correlation between the spectral bands of Sentinel-2. As you can see, we can observe a high positive correlation between the majority of spectral bands with a Spearman coefficient greater than 0.8. So we can see that multicollinearity multicollinearity is likely to be existed between our data. The second criterion used was variance inflation factor. VIF is the most widely used indicator to detect collinearity according to the literature. VIF is given for each one of the predictor variables, ranges from 1, and generally does not have an upper limit. When a VIF is equal to 1, this means that collinearity does not exist between our data, while when VIF is equal or greater than 10, we have collinearity problems between our predictive variables. In the table, you can see the VIF value for each one of the spectral bands of Sentinel-2. Generally, we observed strong multicollinearity problems for the majority of spectral bands from band, especially from band B5 to band B12. The last three criteria that were used was eigenvalue, condition index, and variance proportion. Generally, a smaller eigenvalue close to 0.05 means a greater probability to the presence of collinearity. Another approach is the condition index. Condition index greater than 30 means that we have collinearity between our predictor variables. Moreover, when condition index is associated with high variance proportion greater than 0.50 for 2 or more variables, this means that these variables are significant linear dependent. In this study, you can see on this table, we observed high condition indexes for the majority of the dimension, although these condition indexes were not associated with variance proportions greater than 0.5. So, using these criteria, we don't have a clear indication of multicollinearity as we had in the previous two criteria. So, using Spearman's correlation coefficient and variance inflation factor, we removed the high correlated predictor variables, and the multicollinearity issue was addressed before running the regression analysis. Especially, 5 low correlated variables were selected as the predictor variables of dissolved oxygen. These variables include the spectral bands B2, B3, B4, B5, and B12. Using these predictor variables, the regression model was specified and the regression analysis was run. The significance and the R-square of the model were checked, and as you can see, the model was significant with R-square equal to 0.7. Another important information that we can take from this table is that the spectral bands of Sentinel-2, B3, and B5 seems that are related significantly to dissolved oxygen estimation, while the band B3 seems that have the strongest impact on it. In this study, we investigate the use of remote sensing as a tool for the estimation of dissolved oxygen in marine aquaculture. For this purpose, we used in situ dissolved oxygen measurements and satellite data to conduct a regression analysis. In order to address the collinearity issue between the spectral bands of Sentinel-2, we use 5 correlation criteria. Based on Spearman's correlation coefficient and VIF, the multicollinearity problem was remedied, and so 5 low correlated spectral bands, B2, B3, B4, B5, and B12 were used. From the results obtained from the model, the spectral bands B3 and B5 seems that are related significantly to dissolved oxygen, while the band B3 has the strongest impact on it. So, closing this presentation, I would like to say that the data set selected in this study was only used for the training of the model, and more data needed in order to validate and test the performance of the retrieval model. Thank you for your attention. Thank you very much.
Thank you very much, Ms. Dimouli. I don't know, are there any questions from the audience? Okay, I would like to make a question.
Yes.
I'm not sure if you mentioned it or just failed to understand. The measurements of the dissolved oxygen in the sea. What was the depth of it? And
surface, 0 meters. 0 to 5 meters. Yes, yes.
Okay. Then that makes me ask something more. Do you planning to use to make a model, or I don't know, somehow to give an estimate in greater depths that is all oxygen, or?
Yes, the next step is to research this. For example, we investigate the 10 meters. Yes, the 10 meters. After surface, we investigate the 10 meters.
Okay.
Because it's the maximum depth of the fish cages, or from the nets of the fish cages.
Exactly. Thank you very much. Very good.
Thank you, thank you.
So now let's move to the next presentation. The title is Effects of Fish Meal Replacement by Different Insect Meals on the Nutrient Digestibility and Proximate Composition of Gilthead Sea Bream Sparus aurata. The presenter, Mr. Petropakis. I don't know if it's Mr.
Yes, yes. Hello. It's correct. Petropakis, Theodoros.
Okay. Are you ready?
Yes, yes. You can see the slides.
The floor is yours. Okay, perfect.
Okay. Yes. So, hello everyone. Thank you very much for attending my presentation today. I wish I would be able to attend at Mytilene, but unfortunately I didn't make it. So the project I'm about to show you today is about examining the effects of fish meal replacement by different insect meals, and showing the effects on nutrient digestibility and the proximate composition of the Gilthead sea bream. So first of all, I would like to tell you some notes about why we're trying to replace fish meal by insect meals. Insect meals are gaining much popularity nowadays in aquaculture feeds for several reasons. One of them, although insect meal farming is a heavily industrialized process, as you can see, it's still very energy intensive, and we cannot say it's 100% eco-friendly. The thing is, we can have very efficient land utilization, meaning we can have a small surface area producing a lot of feed. And the main point of using insects is that their production gives value to otherwise unutilized energy-rich waste streams, which guides towards a circular economy scheme, as you can see, which is highly valued in terms of sustainability. Also, insects provide a rich supplementary source of high-quality nutrients, including proteins, vitamins, minerals necessary for optimal fish growth and health. Lastly, in the Mediterranean, climate conditions match the criteria for the culture of certain common insect species, which is also a great opportunity for focusing and specializing the economy, which is also a nice way to sustainably grow as a country. So what is sustainability? Ultimately, sustainability focuses on meeting the needs of the present without compromising the ability of future generations to meet their own needs. Sustainable growth ensures that natural resources are conserved and maintained by supporting a growth model with minimal environmental footprint, and while also being socioeconomically viable. In a world which undergoes a rise in population and increase in demand for high quality protein diet, including fish, it is also has to leave room for further development. So insect meals can be a great way to sustainably grow the whole aquaculture industry. So let me tell you some peculiarities of the insect meals. They present inadequate fatty acid content relative to the needs of the fish, meaning they don't have any EPA or DHA fatty acids, which is essential. Also, the nutrient composition can vary significantly depending on the culture substrate and the processing methods. Hence, it is quite renowned that defattening methods are required prior to including them in an aquafeed. On the other hand, insect meals contain certain fatty acids like lauric acid, which presents immunostimulant properties for fish, which is also another aspect of a way that we can utilize insects in aquaculture. Finally, the most important peculiarity of insect meals is the high concentration of anti-nutrient factors like mostly chitin, which in significant amounts is shown to decrease digestibility of nutrients and reduce growth. So let me tell you more about our work. So this was part of a broader research project where we chose to focus on 3 insect species: the silkworm larvae, the Bombyx mori, the Hermetia elusens, and the Sophobas morio species. The point was to compare a widely researched and approved by the EU insect species, such as the Hermetia, with novel insect species, such as the Sophobas morio and the Bombyx mori, which in particular, the Sophobas morio has been replaced by other studies in the Gillhead Cyprim. And while the Bombyx mori has not yet been researched in the Sparus aurata diet, although it's widely used in other carnivore fish species, mainly in Asia. Both the Bombyx mori and the Sophobas morio are not yet approved by the EU, but the European Food Safety Authority has made prompts, if I remember correctly, at 2021, that it is safe to include in aquaculture feeds, and so it will be included in the following year's legislation. So as you can see in the table below, the approximate composition of the insect meals shows a wide range of deviation, but there is a high protein content, which is crucial for replacing fish meal, although a defattening process has to be followed. So the main aim of this study was to examine the effects of 30% dietary fish meal protein replacement by the 3 partially defatted insect meals of Bombyx mori, Hermetia elusens, and Sophobas morio, and determine the effects on nutrient digestibility and proximate composition on the diet of the Gillhead Cyprim, on the whole body and the muscle tissue. So the insects were reared in the entomology lab of the collaborating Department of Agriculture, pre-processed for defattening and determining the proximate composition. Then processed with the other ingredients and extruded by a pelleting machine at 1.5 millimeters diameter. We produced 4 diet formulations, one with 25% fish meal inclusion, the control diet, and 3 experimental diets replacing the 30% protein coming from the fish meal, namely the Bombyx mori diet, named SWP, containing 8% of Bombyx mori partially defatted meal, the HM diet containing 10% Hermetia meal, and the ZM diet containing Sophobas mori of 9%. All the diets were isonitrogenous, isolipidic, and isoenergetic. And as you can see, for the insect meal preparation, we dried the pupae at 40 degrees Celsius. We defattened using petroleum ether protocol. We milled and determined the proximate composition. So for the rearing conditions, they were maintained at stable conditions for temperature, dissolved oxygen, total ammonium content, photoperiod, and feeding was daily at 1% of the mean body weight of the fish. The whole experiment lasted 34 days, and feces collecting was done during the last 10 days. The feces that were collected were firstly centrifuged for 10 minutes, then freeze-dried and analyzed for proximate composition and for determining the apparent digestibility coefficients. It is noted also that we contained an inert marker in our feed formulation. Didn't point it out very much, but we had 1% of chromium trioxide in every diet. So yes, this was our culture system here, our 500-liter tanks. Gross energy was determined using an adiabatic calorimeter. Fat content approximate was done via Soxhlet petroleum ether extraction. Crude protein approximation via the Kental method. The ash via the ashing in a muffle furnace, and moisture content via comparative weighting. So let's move to our results. The apparent digestibility coefficients for protein was highest in the SWP group, which was the silkworm, as I have put it here also. It presented significant differences between the control group and the other two insect meal groups. As for the apparent digestibility for the lipid, we also had significant differences between all groups, with the control group having the highest digestibility. Please remember this pattern because it's also reflected in the proximate composition later. As for the gross energy digestibility coefficient, we also had significant differences between the control group and the experimental diets, but the highest coefficient was for the experimental silkworm diet, which is interesting. Let's move to the results for the whole body and the muscle tissue proximate compositions. For the whole body, we didn't have many differences. Only in the crude lipid content, where we had statistically significant differences between the SWP group and the Zophobas group with the control. The Hermetia wasn't significantly different from the control. But for the muscle tissue, we detect a pattern where the insect meal diets presented higher values for crude protein content, most probably because of the reduction in lipid deposition, and also in combination with the physiological characteristic of fish maintaining a stable content of protein in their tissue. For the crude lipid content, it was found to be significantly higher in the control group, and there were significant differences with the experimental diets, which reflects the pattern for the digestibility coefficients, if you remember before. So, yes. This can be most probably attributed due to the reduction of lipid digestibility caused by the heating content. The heating is shown to reduce bile salt production, so we have to do more research in that part. Concluding, by our results, we can tell that there are notable differences when substituting 30% of the fish meal's protein content by the 3 insect meals diet. With a partially defatted silkworm meal diet being superior to the fish meal in some aspects for the gross energy or for the protein digestibility factors, which is interesting, whilst the Hermetia and the Zophobas diets seem to be inferior to the fish meal diet. Although still insect meals seemed suitable for inclusion in the Cibrim's diet because we still had good fish growth indices, which were presented by previous studies on the same experiment. We need to do further research on the qualitative changes, effects on the microbiota changes, and on the more special ways which affect the nutrient deposition. But we can tell from our results that we may need to do further defattening to follow a more thorough defattening process, maybe to exclude totally the lipids contained in the insect meals, and also very important to consider ways to reduce the undietary factors, like the heating, because it seems to be the greatest impact factor. So again, thank you very much for attending my presentation. I will be more than happy to answer any of your questions now. Feel free to connect with me or any one of our great team. I would also like to thank a lot my professor, Mr. Karpanagiotidis, and his team for helping with everything. Thank you very much.
Thank you very much for the presentation, interesting presentation. Do you have any questions from the audience? Yes, the microphone.
Hello? I want to ask you something. The insect food has a great potential, but there are some problems about how you can use the whole quality, the quality protein, because maybe we have 80 or 90%, but we are not sure how which of this amount can fish make as a growth rate or energy rate or anything else. So if you can go to a slide that you have got the approximate composition of the food. Is it easy?
Yes, yes, let me share. The presenters have to make me co-host again because I cannot share my screen. I'm waiting to become a co-host again.
I'm sorry for the double, by the way.
Okay, I can share it again. Yes, let me show you. So this is a good point about the protein content, of course. If you mean this approximate composition.
This and one other slide after that one. I want to ask, how do you calculate those percentages? In which way? And if those percentages are the ones that fish can use immediately, or if there are any loss of the protein through these percentages. I mean, for example, the percentage is written at 80%, but the fish can use only 40 to 60.
Of course, yes. This is about bioenergetic mostly. The approximate composition here is determined via the Kendall method. So it is in no way the amount of protein that fish can utilize and deposit in the tissue, of course. The approximate composition here is for the feed, and later on, the approximate composition here is for the tissue of the fish. So of course, we have the initial energy of the feed, and then we have the metabolizable energy, which is actually the retainable energy, which is the energy that fish retain after excreting their through feces. We have losses of protein through feces, through urine. So if you mean this, how we can determine the and also I will show you the formula, because that's why we use an inert marker, because the inert marker cannot be digested. So let's say we have 10 kilos of feed with 1% of marker in its feed, and then we collect 5 kilos of feces, and we find the same 1 kilo of inert marker there. It means that we had 50% of digestibility efficiency. I don't know if I helped to answer your question.
Yeah, I'm covered because there is a great potential at that, and because of my experience in the aquaculture sector, because I worked in the production. We are very stubborn in the food production, food issues. If there is no fish, we do not use. That's the problem. So solutions with great potentials, such as insects or plant-based foods, are very good ideas, but we have to make the optimum the best thing that we can do from them. So thank you for your time.
Yes, no problem. I would also like to add, like this is about the FCR, which is the feed conversion ratio, meaning we feed the fish 5 kilos of feed and we get a growth of, for example, 1 kilo in the feed. This is about the feeding efficiency. That's a great point. Yes. Thank you very much also.
Anyone else? Yes. Just be quick. Hello, Theodoris.
Hello.
How are you? Okay.
Hello, Mr.
Cool.
Condilator.
I have this thought. Maybe you have some answers, or maybe you also have some thoughts. This the use of insects produces wastes, or maybe unwanted parts like the excess fatty fat and the chitinous parts of the insects. Have you any thoughts about what you can do with them?
There are many ways to reduce the heating content. First of all, we don't use the insect at its adult stage. We use the pupae, which is an earlier stage of the insect life, which shown to has the least concentration of heating. The ways that heating can be reduced is via certain enzymes like heatinase processing, and I believe there are ways to also retain the energy loss from this process also and use it for agricultural ways, or to retain the wasted energy also. So, and the fats as well can be used in other sections of the food industry, like for human consumption or for
Okay. So
livestock.
they're not completely waste. There is they are usable, eh?
Of course. Yes.
Yeah. Okay. Okay. Thank you very much.
Goodbye.
Well done. Bye. Let's move now to the next presentation. The title is Effect of Starvation on the Body Weight and Body Composition of Meager Argyrosimus regius at the different water temperatures. Mr. Naumann, I'm not sure for the name. Naum. Yes.
Good afternoon, everybody. I hope so. It's visible.
The sound, you don't sound very good.
Hello. Is it audible at the moment?
What?
Excuse me, Mr. Naumann, we can't hear you very well.
Okay.
In fact, we cannot recognize any word of those you're saying.
Oh, okay. I think I should reconnect it again. That's going to be a better option for me. Good afternoon. Am I audible now?
Excuse me, we still cannot hear you very well. You can test your microphone from the from the left side of your screen. There is a little microphone and the word audio. You can press the arrow next to it and try to test microphone and camera. Maybe this will fix the problem.
Okay. Am I audible at the moment, or not even a little bit?
Your camera is good, but I think that the test.
Okay. Maybe I'm using both of audio and the camera at the same time. That's why it's having an issue.
You're using an earphone, and is there also a microphone on your camera? So you can
Yes.
use. You can use the microphone of your camera and not
Okay. See,
better
am I
now.
audible now?
Yes, better now. Thank you very much.
See, perfect. Thank you. I hope so. The screen is also is visible. My name is Mohammed Noman, and I'm an Erasmus Mundus scholar. I'm currently doing a thesis dissertation in Portugal, but I've done my M1 internship in University of Crete, HCMR, and where I got the opportunity to work on a project which is effect of starvation body weight on a meager under different water temperatures. So the main objective of our study was about physiological changes that a meager undergoes to acclimatize and survive under prolonged feed deprivations. This was a main objective, and also to find out the different adaptations that meager undergoes in aquaculture that could be applied in aquaculture industry by different parameters like growth rates, including SGR, HSI, BSI, and conditioning factor. A little bit of the background of the study that the fish can be moderate availability on different temperatures, and according when it goes to the hunger and it goes to the starvation, it uses the glycogen as a first store, and then it depends on what kind of starvation it is. If it is a short-term starvation, it reacts differently than the long-term starvation. And the fish can also alter their digestive length. Track length also, but we are specifically talking about the meager, which was limited below 17 centigrade and decreased its activity below 13 to 15 centigrade. The temperature impacts the protein metabolism in a fish. It depends on the temperature. Like in colder temperature, the protein breakdown is more rather than at warmer temperature. It synthesizes and it also increases the growth, nutrition, excretion, and oxygen consumption, but may deteriorate at very high temperature. The overall project outline was to find out what temperatures and what kind of starvations at what kind of the body molecules which are using at a particular time, like either it's using lipid, protein, or both at what time of the stages. But technically there are 3 stages of the starvation, which are one of them is very early and which is transient. Second one is a protein conservation and fat utilization, and the final one is the protein oxidation. In material method, we have done this project in HCMR, Hellenic Marine Research Center, and we have acclimatized the fish. It was about 120 gram. Around that, we have around more than 200 fishes at 20 centigrade, and then we divided into 4 different group ranges. At one of them was 17, and then 19, 23, and 26. And we changed the temperature from 20 up and below by 1.5 centigrade each day. And also we run a triplicate and 17 fishes per tank. Every tank has a RAS, so the whole system is the flow system was the RAS, and then it's 1,000 liter per hour. We have sampled on the first day as a control. We have sampled about 5 fishes and 5 fishes per tank on the successive samplings, like on 14 day, 28, 41, and 60 days. The overall project was of 60 days of starvation. It's a long-term starvation. And we have analyzed about the standard growth rate, the HSI, VSI conditioning factor, the carcass fat, and the protein too. Related to the things which we made showed about the how we did it, like what the protein analysis we have done is by the crude protein made with the nitrogen analyzer, and the other things like fat was a Folker's method, which was using the normal procedure. The moisture was by the oven and at 9 centigrade, and ash was determined by furnace combustion at 600 centigrade for 7 hours. Coming to the results, the main part of it. At the end of trial, we have an overall survival rate of around 85.78%. If you see at each of the temperature range, at lower temperatures like 17 and 19 centigrade, the survival rate was more, like about in 17, they have 92.15% of survival, and at the 19, it was 88.24. But at higher temperatures like 23 and 26, the survival rate was around 80. Like in 23, it was 84.31, and 26 is less than 80. And fishes were those which are kept at lower temperatures, like at 17 and 19, and higher final body weight, as compared to those who have a higher temperatures have lower body weights. It is also accompanied by substantial decrease in total lipid across all temperatures. And we have done also the as indicator of mineral content as which is ash concentration, which also shows a notable rise on the 14-day, but not that significantly affected by the water temperature shown in the results. Coming to the very first thing, the results of weight. We have done a two-way ANOVA in that. As you can see, the temperature is both temperature and starvation duration are significantly influencing the weight, and their interaction is also affected by the weight loss. Which means that in the first 2 weeks, the starvation period of the weight of the first fishes was similar. But if you see after the form of slope, which is fitted with the linear model, the decrease in the weight at temperature 17 and 19 is not as sharp as at higher temperature, resulting like if you see a number of it, that the weight loss at 13 to 15% at 17 and 19 centigrade of temperature, as compared to 26 to 28% of weight loss overall at higher temperature, which is 23 and 26. Coming to the standard growth rate, it's showing somewhere similar results, which is not as different from that one, that at lower temperature, it's not decreasing not as sharply as at higher temperatures. And you can see that at 17 and 19 are significantly different from 23 and 26, even though at lower temperatures and lower times. Coming to HSI, we have done also two ANOVA, which revealed that the temperature and starvation duration are significantly influencing these. And about till the 14th day, about 50% of the drop in HSI was recorded. And we can see the fish mobilizes the fat from liver, which more intensively at higher temperatures. Higher temperatures, they mobilizes more rather than lower temperatures, reaching to a decrease in 90% till the 60th day. And the fact is exponential model, so you can see that how fast the fat is mobilized in the temperatures, like at higher temperature rather than lower temperatures. And we can see that 17 and 19 centigrade temperatures are significantly different from 23 and 26, but they are not as significant different from each other, like 17 and 19 within each other. VSI, as you know, the meager is not having as much of visceral fat, perivisceral fat. So we have done this without the liver, the VSI, and it's greatly affected the liver weight. But we see the visceral stomach index without the decrease in weight was not very prominent, and there is not a particular pattern in it either. And the decrease in the weight was not very prominent. A shrinkage of the organs was expected then in person, and but at higher temperatures, yes, body reserves are used, but not as much as we expected in that one. In carcass fat, it's a falling exponentially. Like at higher temperatures at 26, if you see it was decreased at the end of 6th day, like 50%. And but the rest of the weights, the first few at higher temperatures, it falls in the first 14 days, but the rest of the temperatures also falls. The carcass fat also falls from 28 days to onward. And coming to the last part of this thing, that carcass protein, which you can see that it is not showing as much of a pattern as we expected, but it is there was a decrease in the carcass protein over time. In 17 degree, the decrease is not as significant, and 19 also, but we can see there was starting from 14 to 28, there was a decrease in the carcass protein. We can see that it might be possible that fish in the higher temperatures are further stages of starvation with protein oxidation more rapidly. And through a model, we can see that how many days it is required for them to use that kind of protein. And if you see at the two-way ANOVA, at 14 day, at each temperature in 14 days, there was not significant or significant different from all of other ones. So in conclusion, we can see that there was understanding of the fasting period and the effect of fish important for the construction of bioenergetic models. Every fish has a different model of things. So we actually concluded that thing that if every fish is using different kind of molecules at different kind of stages, that was the whole purpose of this study. And also shows like the gilthead sea bream can lose fat stores in key areas like muscle and liver without impacting the fillet yield, which was mainly used when there are some industry, the people in the industry has not that much of money to put the feed and put on feed management. And so they can use these kind of methods to not impacting the fillet yield, but keeping the economics of the whole flock at a minimum cost. And fish species react differently to the fasting, and impacts of tissue compositions and overall nutrition value is different. And for example, the meagre, as I mentioned before, that has perivisceral fat, so the main fat mobilization site is different, like it's liver and followed by the muscle. So this was the whole study we have done. I want to acknowledge my supervisor, Dr. Stavros, from HCMR, and Dr. Maria Mustaraki from that, who helped me in doing that. And I would like to thank for all of the things you have done for me. And I would like to answer if they have any questions left for me. And the other efforts, if you have any.
Thank you. Thank you very much. Do you have any questions from the audience?
Hello, very impressive work, and I have to say that I'm thrilled that you used the migra as the key subject in this research, because it's a species with very big potential. So I want to ask you a question. According to this study, right, in my previous job in Corinth, we used to breed migra, but there was a very serious problem. In June, July, and August, the water temperature was way out, about 23 to 24. In August, the water temperature reached at about 30 degrees.
Yes.
No one couldn't imagine this problem. Symbiotic species like sea bream and sea bass weren't fed at all. According to this study, which would be a strategy for mig breeding at those temperatures?
At those temperatures, I don't think so. If the climate change is affecting those kind of things, I don't think so. Breeding will help in that way at higher temperatures, like 30 with this much of humidity. But at lower temperature, if you see at the 26 temperature, it is affecting too much. Even though their fat, their mobilization, the protein breakdown, oxidation is also happening. But at 30, I have no idea that it might be affecting anything left in that thing. But I think it can be useful to look into it. We at first we thought that we were gonna put 30 temperature, and we started with it. We started run the project with 30, but we had a lot of mortality at that temperature. So that we thought we're not gonna continue with the 30 temperature, and we dropped it to 26. That's the reason that even in the pre-trials, it was not sustainable by the fish itself, even though the fish was 110 grams. So I have no idea that what it gonna be, but it is not gonna be very much sounds good to listen. That will gonna happen at a higher temperature of the fish. Hopefully, I answer the question.
Yes, I'm covered. Thank you.
Okay, thank you very much. We're running a bit late. Let's move to the next presentation. The title is The Effect of Insect Meal, Insect Frass, and Hydroponic Byproducts on Growth Performance of Common Carp, Cyprinus carpio, co-culture with lettuce, Lactusa sativa, in closed-loop aquaponic system, AWARE. Okay. Mr. Stamelos, or any other member of the group, of the research group?
Yeah, I'm here. Can you hear me?
Yes, yes. Please start your presentation.
All right. One moment, please. Can you see the presentation? Can you hear me?
Yes, please go on.
All right. Greetings, my name is Alexios Tamelos. I'm a PhD student at Aristotelou University of Thessaloniki. I'm delighted to present to you our study, the effects of insect meals, insect frass, and hydroponic byproducts on growth performance of common carp in a closed loop aquaponics system. Aquaponics is a near zero discharge system that offers not only economic benefits from both fish and plant production streams, but also significant reduction in both environmental noxious discharges from aquaculture sites. Aquaponics could be a controlled environment involving the cultivation of plants, breeding of aquatic animals, in a closed-loop water recirculation system. A CLA system is characterized as a symbiotic relationship among fish, plants, and bacteria. Fish produce ammonia through waste excretion as well as through respiration. Plants cannot directly utilize the ammonia, thus nitrification becomes essential in an aquaponic system to convert ammonia in the forms of nitrogen that plants can absorb. This conversion occurs within the biofilter, where specialized aerobic bacteria transform ammonia into nitrate. The resulting nitrates are conveyed to the hydroponic area of the CLA system. In this area, plants thrive by absorbing the available nutrients. The aim of this study is to evaluate the effects of the diet of insect meals, plant residues, insect frass, and hydroponic byproducts as a replacement of fish meal on the growth performance of carp and on growth and nitrogen uptake of lettuce in a laboratory CLA system. A total of 297 carps and 108 lettuce were distributed in a 9 CLA system. The CLA systems were divided into 3 groups. The C group fed with a controlled diet containing 50% of fish meal. The B group fed with Hermetia diet, where the fish meal protein of the controlled diet was replaced at 50% by Hermetia meal and plant residues. And the F group fed with plant residues, where the fish meal protein of the control diet was replaced by 50% by a mix of Hermetia meal, Hermetia frass, and hydroponic byproducts. The fish tank of each of 9 autonomous CLA systems had a volume of 144 liters. Filtration unit of each system was separated in 3 different parts, including the mechanical filter, the biofilter, and the pump segment with a UV filter. The total cultivation volume in the hydroponic sector was calculated at 2.42 liters. Over the hydroponic session, we installed LED light pumps to mimic natural daylight conditions. Here we can see the system in day 1. You can see the hydroponic area, the fish tank, and behind the fish tank there's the filter with a UV filter here. Throughout the acclimatization period, fish were fed ad libitum 3 times daily with commercial pellets. After this period, fish were anesthetized for initial measurements. Plants were also divided. Plants were also added into the CLA system in day 1. Throughout the experimental period, fish were fed ad libitum 3 times daily. The experiment lasted for 45 days. At the end of the experimental period, morphometric measurements on plants and fish were recorded. Throughout the experimental phase, levels of ammonia remained constantly at 0. All other parameters showed no significant variation across treatments. Carp growth performance exhibited no statistical significant differences among treatments. The average initial weight of fishes was around 4 grams, and the final average weight was recorded at around 8 grams. No fish mortalities were observed during the experiment. Initial measurements of plants indicated an average leaf number of 5 leaves, and plants were weighted around 1 gram. Final average key matrix of lettuce cultivated in different treatments are shown in this table, providing an overview of lettuce growth performance after 45 days in the CLA system. Lettuce metrics showed no statistical differences across treatments. Here we have the nutrient composition of lettuce leaves. Again, no statistical significant differences across treatments were observed. And according to Bryson and Mills' handbook, we may have some deficiency in manganese and iron in our plants in this study. In all 9 CLA systems, lettuce was effectively cultivated with optimal root to shoot ratio and the relative growth rates, which indicated that the plants were provided with enough resource to grow successfully. Tsumalakou et al. Co-cultivated lettuce and red tilapia in a CLA system where iron and potassium supplementation were used to evaluate the growth performance of lettuce. In this table, we can observe distinctions in the nutrient composition of lettuce leaves between the 2 different studies. This concludes that the iron supplementation is essential in the diets of our study to improve lettuce growth performance and nutrient composition. Thank you. Any questions?
Thank you very much for the presentation. Question from the audience? No, this time. Okay. All right. There are no questions. I don't know if there are any questions from the online. Έχουμε ερωτήσει από το... Έχουμε ερωτήσει από τους διαδικτυακούς, όχι. Okay, thank you very much. Thank you. And with this...
You're most welcome. Thank you.
And that was the last presentation for this session. Thank you all for the audience, for the participant, and see you later.